Hello, my respected professor and GAs. I'm here to present my oral presentation on experiment 1, which is monitoring of microorganism growth. So microorganism has been using as experimental models for various scientific researches. And here in this experiment, the growing of baking yeast is monitored and documented by sugar fermentation. In this experiment, a simple experiment of involving the yeast catalyzed fermentation of sugars has been carried out. When yeast becomes active as it breaks down sugar, also known as sucrose, through a process called fermentation. The experimental model for this experiment is warm water, sugar, and yeast for monitoring of microorganism growth. So for the theory, fermentation is a chemical process of breaking down a particular substance by bacteria, microorganisms, and yeast, where yeast strains are actually made up of living eukaryotic microbes that contain cells with the nuclei. And here, as we all know, that is come to life as it breaks down sugar, that is also known as sucrose, which the process is also called fermentation. And here, fermentation is a process adopted by anaerobic organisms to obtain ATP without the use of oxygen. And sugar fermentation results in the production of ethanol and carbon dioxide. In the case of sucrose, the fermentation reaction is as followed over here. And here in this experiment, warm water and sugar activated by yeast by the sugar fermentation. Now the experimental materials for this experiment, we need 2 glass of measuring cups and spoons. We need first of all yeast, 2 tubes, granulated sugar of 1 or 3 tablespoons, small clear container, warm water of 1 and 4 cups. Now the flowchart representation of the procedure. So first, 2 plastic clear cups were filled with 1/4 cup of each warm water. In each cup, we added 1 teaspoon of sugar were added and dissolved into the cups with the warm water. After that, 1 tube of yeast was sprinkled into the sugar solution of each cup, and it was observed the change of yeast every 15 minutes time interval, and it was observed 4 times over 0 minute, after 15 minute, 30 minute, and 45 minute in 2 different environment. Here, in the first container, it was lifted to a light at room temperature, and it was observed the change of the yeast every 15 minutes interval for 4 times. In the experiment, the second container was wrapped with a plastic and it was placed in a warm temperature, as called a different temperature. The change of the yeast was observed every 15 minutes in the same manner. So now here is the data presentation. So the first picture is for photo taken at time 0 minute. Here, just after dissolving the yeast with the warm water and sugar, yeast just started to dissolve in warm water solution. After 15 minutes, there was a formation of thin layer of foam bubbles. And here, the picture is after 30 minutes. Here, the formation of foaming layer, kind of 2 centimeter, is increasing by the time. And after 45 minutes, it was eating up the sugar by fermentation. As we can see, there is a kind of bubble of foamy formation. So foam rises. It was rising a little bit higher than 2 centimeter after 45 minutes, and it was creating a lot of bubbles. And these pictures are for a different environment, which was carried out in a warm and sealed environment. So I sealed the cup with a plastic, basically a transparent plastic. And the first picture is taken at 0 minute. So the yeast started to dissolve in warm sugar water solution after the sprinkling. And the second picture is after taken 15 minutes, and it started to produce thin layered foam. Then here the picture is taken after 30 minutes. It is producing more foam than the clear plastic is rising upwards because it is producing gas inside. And the fourth picture is for after 45 minutes, where foam rises by the time, and the plastic is expanding at because there is a gas inside and it's creating pressure. After some time, the layer disappeared. So for the observation and conclusion, I would say the yeast was able to act differently in both conditions, basically because each glass was placed in a different environment, the two glasses. The foam formation is the result of yeast using the oxygen within the air digesting the sugar, and the production of a byproduct gas, which was carbon dioxide. So basically in the first glass, oxygen is quickly available as it was open, and it was added to the sugar solution. So it began to chemically convert the sugar within the water and oxygen within the air into energy. And here in glass 1, it was activated by adding warm water and sugar. Basically the yeast was activated, and the foaming that results from the yeast eating up the sucrose is the cloudy appearance formation. And the glass 1 also smelled very differently. And after that, for the second glass, it was wrapped and placed in a hot temperature. There was not oxygen readily available as it was sealed glass, and as there was no oxygen, the waste products of this chemical action were different, and it leads up to formation of carbon dioxide, by which the plastic was expanded and there was a pressure. So by the monitoring time, the experiment slowed down in the sealed cup, and there was less energy produced, and ethanol produced is really toxic for that. So the yeast cell starts to get damaged by slowing down the growth, so the layer disappeared. And now I will talk about the similarities between the home test and the laboratory experiment, that for the experiment 1. So there are basically various similarities in between this home test and lab test. So I will say for the spectrophotometer in the lab analysis, instead of LV liquid medium, warm water has been used in here in the home experiment, which is the food for yeast. Instead of bacteria that we can use in lab, yeast has been used. Instead of spectrophotometer, we have eyes and light bulb to visualize. The solution can be lifted and put under the light to visualize how turbid the solution is to make the conclusion of yeast growth. And in lab we have cuvette, and here in home test I have used a plastic container, and 2 different plastic cups have been used with warm sugar water solution with yeast in terms of reference with the sample and other sample in terms of the lab experiment. So that's all for my presentation. Thank you very much, and thanks for your cooperation.